Hello everyone, hope I am audible. So let us begin today's class. I assume my screen is visible. Yes, my screen is visible, right? Okay. So let me welcome you all to the week 7 PMRF-NPTEL session on the NPTEL course Introduction to Developmental Biology. My name is Anuvab Chatterjee and I am a PMRF fellow and PhD scholar under the care of Professor Jonaki Sen at the Brain Developmental Biology Lab, Department of Biological Sciences and Bioengineering, IIT Kanpur. So, as you have, if you have seen the previous videos, you already know. But if you don't, PMRF NPTEL sessions are designed in order to interactively solve problems and assignment questions based on the particular NPTEL course, so that the students who are taking the course itself understand the various concepts behind the assignment problems, as well as have a better understanding of the week's information and the courses that and the lectures that were delivered. In this particular week, we are going to look into Drosophila axis development and various lectures on that, and we will be solving the previous year's questions of this particular week in order to understand various concepts behind Drosophila axis specification. So let us begin. So we'll start right off with the first question, which is, in the process of cell differentiation, the term determination refers to a state in which the cell is. So there are four options which include: it is capable of autonomous development even when present in a different context. It is capable of autonomous development only in a neutral context. It looks morphogenically different from its undifferentiated state, and it is still amenable for switching to different fate if appropriate signals are present. So, for a bit of background, during cell differentiation, as various progenitor cells after dividing, they undergo the process of differentiation in order to form specific lineages. For example, the colony forming unit cells in our blood, which are the blood progenitors, give rise to various blood subtypes such as red blood cells, white blood cells, as well as the platelets. This is known as the process of differentiation, in which a progenitor is specified to undergo cell divisions and is now turns into a cell which will only have one fate determined to it. That is, after subsequent divisions, the one particular daughter of the colony forming unit cell, the CFU, will form the red blood cell only. This is the process of cell differentiation. It essentially has two parts. One of the parts includes the specification stage, and the second part includes the determination stage. And the question itself asks if we know what exactly the determination step refers to. So the options are: the term determination refers to a state in which the cell is capable of autonomous development even when present in a different context, capable of autonomous development only in a neutral context, looks morphogenically different from an undifferentiated cell, and is amenable for switching to different fate if appropriate signals are present. So I would request anyone trying to answer this. So Muliani says it is A, that is, it is capable of autonomous development even when present in a different context. Okay, so we'll try and look into what the solution slide says on this. So as I said, differentiation involves two particular steps which are known as specification and determination. So commitment is also a concept that comes under the umbrella of differentiation. Commitment is when the cell has fate specified to it that it will only form a particular type of cell. Now, commitment has two subparts, which include specification, in which it is capable of autonomous development, but only in a neutral context. So, you can consider it as given, say, the example we are taking, the colony forming unit can either form a red blood cell or say a white blood cell. If at the stage of specification the colony forming unit cell is left alone completely, then if its fate was already determined to be an RBC, it will go ahead and form an RBC in a neutral context when there are no neighboring signals coming to it to form some other type of cell. If there are neighboring signals coming to it to say it is being asked to remain as a progenitor or change its fate to form a white blood cell or a platelet producing megacardiocyte, then it will go ahead and do just that. So in case of specification, the cell can still be asked by neighboring cells via various signaling pathways in order to form in order to change its particular fate. But in case of determination, which is the next step after specification, various other various other developments have already taken place inside the cell. Various other gene expressions have already taken place, so that even when there is a neighboring cell interaction which is asking the cell not to undergo its specified fate, it cannot go back. It has to differentiate into the specific fate it was determined to have. So if it was going to form an RBC, even if we take it and ask it to form a WBC, or if we put it in a completely different context in which it is getting various signals to form, say, neuronal cells, it will still go ahead and form an RBC. So in the determination step, even the cell is capable of autonomous development even in a different context. So context is something that we will look into in later slides as well. But if we go back to the previous example of context based differentiation, if you remember the diagram we had seen in which there was a mesenchymal layer on which we were transplanting different parts of epithelia from different parts of the body, we were able to see that the mesenchyme was able to produce various signals that was asking the epithelium to form various different characteristic features of the mesenchyme itself. So in the context of a particular mesenchyme, the epithelial cell's characteristic was changing, and that is what we mean by context. Depending on the position in the embryo or the neighboring cell itself, a cell might undergo various different stages of development and can have various different fates. But once the cell has reached the determination stage, even if it is asked it is given different signals, or it is completely taken out and transplanted to a different context, it will still form the fate in which it was determined. Thus this is specification and determination, and during these stages the cell might not look differentiated even though its developmental fate has become restricted. So various of these changes can be changes in the gene expressions itself. So we do not see a outward phenotype on the surface of the cell itself, so that we understand that it has become a particular type of cell, but its fate has already been determined. So in the process of cell differentiation, the term determination refers to a state in which the cell is capable of autonomous development even when present in a different context. So is this clear, the difference between specification and determination? Anyone has any questions regarding this? Okay. So we move ahead. The next question is, which of the following is or are the main conclusions from Dreiser's experiments on the early development of sea urchin embryos? The options are: each cell carries only genetic material required for its development. The fate of a cell is governed by its embryonic relocation. Cell has more developmental potential than what normally develops into an embryo. Only the germ cells carry the genetic information to the subsequent generation. So if we remember what exactly Reich's experiments were, what Reich did is that he experimented with sea urchin embryos, in which he tried to replace the neighboring cells, but the connection between neighboring cells, so that the particular cell, if it had say a neighbor A and B, it now had the separate other neighbors C and D, and he tried to demonstrate that if this particular cell will develop normally, or it will the cell will develop depending on the context of the neighboring cells. So these were the experiments Reich did. So the question is, which of these following conclusions are can be developed from Reich's experiments? That each cell carries only the genetic material required for its development. The fate of the cell is governed by its embryonic location. Cells have more developmental potential than what it develops normally into in an embryo, and only germ cells carry the genetic information to the subsequent generations. So I would ask everyone to try and answer this. Brian says the answer is B and C. Fate of the cell is governed by its embryonic location, and cell has more developmental potentials than what it normally develops into in an embryo. Anyone else wants to try and answer this one? This is a box type of question, so it is more than one correct. So Sangeeta says, oh, it is B. The fate of a cell is governed by its embryonic location. Okay, anyone else? Okay, so let us look at what the experiments were. So this is the sea urchin embryo as seen from the top. So if we see that we have two poles, the animal pole and the vegetal pole, and on top of there, from the top we can see we have the cells 1, 7, 5 and 3 present in the animal pole, 2, 8, 6 and 4 are present in the vegetal pole. So this is a side view of the same. After a single cell division, we have the various daughters of one labeled 1A and 1B, and similarly for 2, 2A and 2B. And we have arrangements such as this, in which the vegetal pole cells are together. So you can see that 8A and 6B has 8A is connected to 8B and 6B as well as 7A. Similarly, 5B is connected to 7A, 5A and 6B. And thus the connection between the cells or the neighbors are very highly specified. So this is what in normal cases what the division would be, and these are the neighbors the cells would have. So during Reich's time, what was believed is that majorly believed was in autonomous development. That is, this particular cell 1A would say if we lineage trace it, it will if it forms the ectoderm, then even if we change the location of 1A, it will still form ectoderm. Thus, in order to demonstrate it that the developmental potential of a cell does not depend on the neighboring cells, what Reich did is he took a glass plate, and during this stage he pushed down the cells together on top of each other so that the cells get squished, and we do not have a very distinct animal pole and vegetal pole cells separated out. Instead, we have that the cells get like the cells go, if they were like this, they now go in between each other, so that now the connections get completely disrupted and the neighboring cells are completely different from what it previously was. So if we can see here now, 6 is connected directly to 7 and 5, whereas in the normal one it should have been 8 and 4. And now after the first cell division was done, he removed the glass plate. So after the 8 to 16 cell stage division was done, he removed the glass plates and allowed the embryo to develop normally. What he expected is that since he believed that these particular cells will now grow they were already determined to grow at the 8 cell stage, so one should have formed ectoderm or whatever lineage it should have formed in this particular position as well, in the new position. So the straightforward conclusion from this is that the embryo that will develop will be deformed completely. So it will have its heart on the other side, its hands on different places, because the what he believed is that the cell should form whatever organ it was about to form, or whatever cell lineage it was about to form, and it won't be determined by its location. So location changing by this particular experiment won't change anything else. But after allowing the embryo to develop, to his surprise, he found that the embryo developed normally. And these were the conclusions from Dresh's experiments, that since the embryo developed normally, then whatever cell now took the place of one. So, say if number two got in after the rearrangement got in place of number one, cell number one, then cell number two will give rise to ectoderm. Even if in the normal embryo it wouldn't have had formed the ectodermal lineage. Thus, what he concluded is developmental potential of a blastomere is greater than what it develops into as part of a normal embryo. So the cells 1, 2, 3, 4 could all have developed into ectoderm, but in the normal embryo they develop into whatever their particular embryonic location wants it to develop into. In normal development, the neighboring cells prevent it from generating all cell types a given blastomere is capable of. So what it means is that the cell number one will only form, say, ectodermal lineage of cells and not form any mesodermal cells or endodermal cells. Or if one formed, say, the eyes, the lineage finally formed the eyes and the optic cup. It will only form those cells and not end up producing cells which are similar to the cells in the heart. It is only because the neighboring cells prevent it from forming anything else and specify its lineage. And finally, the fate of a cell depended solely on its location in the embryo and nothing else. So, the location is what matters entirely. And thus, the conclusions from Reich's experiments on early development of sea urchin embryos are: the fate of a cell is governed by its embryonic location, and cell has more developmental potentials than what it normally develops into in an embryo, but the neighboring cells allow it only to form a particular lineage. So, not in this context, but in another context, we have already seen it. In the C. elegans vulval development, we have seen how the the P6 cell does not only undergoes primary fate, and it signals to the P5 and P7 cells to undergo the secondary fate. So the neighboring cells actually determine what the particular cells will develop into. So it is not just that one cell can autonomously develop throughout the embryo, but the neighboring interactions are also very important. This is what the main conclusions from Reich's experiments are. So anyone wants to ask anything about this particular question? Okay. So which of the following results of Nuslin-Volhardt's experiments provide the strongest support for morphogenetic activity of becoid? So Nuslin-Volhardt is a Drosophila scientist who actually won the Nobel Prize for designing the Heidenberg Screen, in which she actually showed the world how exactly to screen for various mutant phenotypes. And that was a very great work. If you can just go back and read the book about Scott Gilbert, you will understand what exactly the screen was developed for. So exactly is so which result of her experiments have provided the strongest support for morphogenetic activity of becoid? The options are: becoid mutants do not form anterior structures, injection of anterior cytoplasm for the wild type egg restored normal anterior development in becoid mutants, becoid concentration was highest in the anterior and lowest in the posterior, injection of wild type anterior cytoplasm to the middle of becoid mutant embryos led to the formation of thorax on either side of injection. So for a bit of context again, what is a morphogen? Morphogen are genes or compounds or chemicals, various genes whose activity depends on its concentration, or how a particular cell will react to its to the particular morphogen depends completely on the concentration of the morphogen. So if the concentration is very high, then a cell will undergo one fate. If the concentration is very low, the cell will undergo another fate in the presence of the particular morphogen. So the question asks us what results of Nusslein Volhard's experiment provide the strongest support for morphogenetic activity of bicoid. So what exactly proves that bicoid is actually a morphogen? And these are the four options. There is only one option correct because it is a round type answer. So if anyone wants to try and answer this. Okay, so no one remembers, it seems, the experiment. Mullany says A. Bicoid mutants did not form the anterior structures. Okay. So someone wants to add something? Okay. So let us look at the slides itself. So as I said, morphogens are gradients or proteins in to which the cells must respond to it. So what exactly are the characteristics of morphogens? Cells must respond to the particular protein, and differentiation would depend on its concentration. So depending on the concentration, it will undergo a particular fate. So what exactly happened in bicoid mutants? So we in the A box we have the particular normal wild type gradient of bicoid. So as we can see, it is maximum in the anterior portion, and the concentration dies out as we go to the posterior regions. But in a bicoid mutant, we see no bicoid, and there no anterior structures, so head and thorax do not develop. Thus, this so the first part is here. So the first part is there. The cells actually seem to respond to bicoid, and it depends on the concentration of bicoid itself. But it needs to be proved. So what exactly the proof was? So here in the C diagram, we can see that the concentration of in the wild type egg, the concentration gradient of bicoid is like this, where it is maximum in the anterior, minimum in the posterior. When we transplant part of the cytoplasm of bicoid from the anterior portion into a bicoid mutant egg, in the middle of a bicoid mutant egg, so it initially did not have any bicoid gradient, but after transplanting the cytoplasm, it has this huge amount concentration of bicoid in the middle of the cytoplasm, in the middle of the body, and thus we have the concentration gradient such as this, in which in the middle we have maximum bicoid, anterior posterior we do not have any bicoid. So if it is a morphogen, then bicoid must give rise to specific differentiation, like the cells should differentiate in the middle of the body as if they were having the maximum concentration of bicoid, that is they should form anterior structures, as you can see right here. And that is what happens. In the middle of the body we have the head, and on either side of it we have the thorax, and no head develops on the anterior portion where it should have in the bicoid mutant. So we have this particular mutant in which we have maximum bicoid concentration in the middle, and in here anterior like structures have developed. So we have head and thorax in the middle itself. So this particular experiment proves that bicoid is actually a morphogen. Thus, the option is injection of wild type anterior cytoplasm in the middle of bicoid mutant embryos led to the formation of thorax on either side of site of injection. That is, artificial production of the concentration gradient or artificial manipulation of the concentration gradient also gives rise to the specific differentiation, but in different parts of the embryo. So bicoid mutants did not form anterior structures. It shows that bicoid is important for anterior structure formation, but does not show that the bicoid is important for or bicoid is acting like a morphogen, that the concentration gradient part of it is not there. Okay, so we move on. Which of the following is/are the activity or activities of bicoid during anterior posterior axis specification in Drosophila embryos? Bricoid suppresses translation of hunchback mRNA. Bricoid activates translation of hunchback mRNA at the anterior. Bricoid activates hunchback transcription. Bricoid suppresses caudal translation. So these are the four options. This is a box type of answer. So more than one is correct. So what are the activities? What exactly Bricoid does? So anyone has any idea on this? Sangeeta says A. Bricoid suppresses the translation of hunchback mRNA. Okay. So let us look into the slide itself. So this is exactly what Bricoid does in the embryo. So we have already, this is a concentration gradient of various mRNAs that are present in the oocyte, and we have Bricoid mRNA concentration only in the anterior, nanos mRNA concentration only in the posterior. So as Bricoid is an anterior factor morphogen, nanos is a posterior morphogen. And we have these two proteins, hunchback and caudal, whose concentration is equivalent throughout the embryo. So hunchback gives rise to anterior-like features and caudal gives rise to posterior-like features. So these are proteins and these are not morphogens. These give rise to the various differentiation factors normally. Now, in the early cleavage embryo, the protein concentration. So this was the mRNA concentration and this is the protein one. So we see similar effect in Bricoid and nanos concentration. So as the mRNA was only in the anterior portion, Bricoid concentration, protein concentration is only in the anterior, drops down towards posterior. Similar for nanos, it is present maximum in the posterior. What Bricoid does is goes and inhibits caudal, and nanos goes and inhibits hunchback. So we have a similar expression pattern of hunchback and caudal which look just like this, in which caudal is also present in the posterior and hunchback is also present in the anterior. So what exactly happens is Bricoid protein prevents the translation of caudal in the anterior part, and nanos protein prevents the translation of hunchback in the posterior. And this is how they prevent the various anterior and posterior factors are prevented from being expressed in the opposite areas. So if nanos was not there, then we would have an evened out expression of hunchback, and then there would be various axis specification problems. So the options are Bricoid suppresses the caudal translation and Bricoid activates hunchback transcription, as you can see just right here. So let us move on. Next question is, what are ring canals? So ring canals, for context, it is important for again oocyte development, and it is something related to how the oocyte is connected to the various other nerve cells. So the options are, these are cytoplasmic bridges connecting the adjacent follicle cells that surround the oocyte. These are cytoplasmic bridges connecting the daughter cells produced by the oogonium, the fusome structures that connect the nerve cells, or the cytoplasmic bridges that connect nerve cells and follicle cells. This is one type correct answer because it is round type. So anyone wants to try and answer this? What exactly are ring canals? B. Millennium says B. Cytoplasmic bridges connecting the daughter cells produced by the oogonium. Anyone else wants to try and answer this? Arup says D. Cytoplasmic bridges that connect the nerve cells and follicle cells. Anyone else wants to try? Okay. So let us look at it. So first let us look at the oogonium, which is the first cell that is produced by the germline cell, that is produced from the germline precursor cells, which are stem cells. So they produce a daughter which undergoes various cell which will finally form the oocyte, and that is known as oogonium, the progenitor cell. It undergoes cell division to form cell number 1 and cell number 2. which then again undergo cell divisions to form 3 and 4. So but in this particular cell, even if separation of nuclear material is there, cytokinesis does not occur. So the cells are still linked, and this linkage is what the ring canals are. So 1 and 2 have a ring canal between them, and when one divides to form another daughter cell, that is 3, and 2 divides to form 4, they are also connected by ring canals to each other. But since 3 and 4, they are completely separate from each other, they do not have a ring canal between them. So this continues till we reach the 16th cell stage, and we can see that the cells 1 and 2, they have the maximum number of ring canals, that is 4, whereas every other cells have lower than them. This presence of the maximum number of ring canals, so the maximum connection to various other cells cytoplasm, specifies which particular cell which form the oocyte will form the oocyte. So either one or two can form the oocyte, and every other cell forms the nurse cell. So nurse cells are various other cells which help in the development of the oocyte. So in the developed in the developed embryo, you can in the developed oocyte you can see that these are the nurse cells that are present, and this is the oocyte that is very larger in size, that has grown larger in size, and these are known as the nurse cells, and this is the oocyte. So ring canals are the connections between the nurse cells and the oocyte. And these are the follicle cells which are present on the boundary. So these do not have, these are somatic cells, they do not have a direct connection with the germline nurse cells or the oocytes. So what are ring canals? Ring canals are cytoplasmic bridges connecting the daughter cells produced by the oogonium. So this is okay. Anyone wants to ask anything? Okay. So we move on. So this particular question is a large one, and this follows the various events which occurs during the dorsoventral axis development in Drosophila. So what are the various events? So we are given 8 separate events, and we have to align them so that they occur chronologically. So the events are: pipe synthesis is inhibited in dorsal but not ventral follicle cells. Ester is cleaved into its active form. Follicle cells are induced to form columnar dorsal follicle cells. Cactus is degraded. Gherkin is localized between the cell membrane and nucleus. Spatula is cleaved into its active form. Tall is deactivated, and dorsal enters the nucleus. So 8 particular events, we have to align them so that we have a chronological order in which the dorsoventral axis formation occurs in Drosophila. So we have 4 options which are very separate. So anyone wants to try and align them or try to answer which one can be the correct one if you remember the entire dorsoventral axis formation. Instead, let us do one thing. We will go through the dorsoventral axis. Mrinani already has answered, which is C. So she has answered C, which says. So we'll go through the order once we go into the slides. But yes, the C is the correct answer. Very nice. So at least someone has gone through the lectures, it seems. So this is the entire diagram of how exactly dorsoventral specification occurs. So we'll go through it one by one. What happens is that first the nucleus of the oocyte is transferred to the to a dorsal region. So the nucleus travels to this particular region, and then nucleus deposits the Gherkin mRNA between the towards the somatic cells, which are the follicle cells. Gherkin, after getting into the follicle cells via like signaling to the follicle cells via the torpedo receptor, prevents the formation of the pipe protein. So pipe in the synthesis is completely inhibited. But in case of the ventral follicle cells, there is no Gherkin, so pipe is still secreted. Pipe is still produced by these particular follicle cells. So as pipe is produced, a signaling pathway occurs, which gives rise, which produces a particular protein, which goes and cleaves gastrulation defective or GD protein, which is present in the perivitreline space. So this space between the oocyte and the follicle cells is known as perivitreline space, and there are various proteins present in it. Because the ventral cells or follicle cells have the pipe produced, they can the ventral follicle cells can produce another protein that goes and cleaves GD. This produces the active forms of GD, active form of GD, which is again goes and cleaves another protein known as snake, which is active to its active form. Snake goes and cleaves Easter to its active form. Easter goes and cleaves Spatula to its active form. Finally, Spatula in its active form goes and binds to a toll receptor present on the oocyte, the various present on the oocyte membrane right here, and the signaling from the toll receptors goes and degrades the cactus protein. So cactus, what it does is it is holding on to another protein known as dorsal and does not allow dorsal, which is the transcription factor, to get into the nucleus. But the entire signaling process, what it finally does is it degrades cactus, and so dorsal is free to get into the nucleus. And once dorsal gets into the nucleus, it produces various signals which ventralizes this particular region where there was pipe signal. So we have the ventralization of the ventral portion of the embryo, and the opposite is stays in its normal dorsal field. So this is the entire pathway, and it is written in written down in here. So we have the entire thing. So this is what, in short, what it was. Gherkin is produced and is deposited. Gherkin inhibits pipe production in the dorsal cells. Ventral cells produce pipe. Pipe produces a entire cleavage cascade, which produces spatula's active form binding to toll. This produces another signaling cascade degrading cactus. Cactus leaves dorsal. Dorsal goes into the nucleus and ventralizes the entire cell. Thus, if we go back to our question and start aligning, the first option is Gherkin is localized between the cell membrane and nucleus, so 5, followed by follicle cells are induced to form columnar dorsal follicle cells which do not produce pipe, 3. Then pipe synthesis is inhibited in dorsal cells, not ventral follicle cells, 1. Then Easter is cleaved to its active form, spatula is cleaved to its active form, toll receptor is activated, and that was 7. Cactus is degraded, and finally dorsal enters nucleus. So the correct answer is C. So anyone wants to ask anything on this? So this is an entire pathway, and we are just aligning the various steps depending on what exactly the chronological steps occur. Anyone has any questions? Okay. So the next question is, which of the following contributes to dorsal ventral polarity in Drosophila embryo? The options are Gherkin present in oocyte, torpedo present in oocyte, polarized microtubules in the oocyte, or caudal present in the oocyte. This is a box type of answer, so more than one options are correct. Which of the following contribute to the dorsal ventral polarity? So this is what we just discussed. So anyone wants to try and answer this? Okay. So this is something that we just discussed. So but I think we have not discussed exactly where the particular proteins originate from. So as we have seen that in the particular previous diagram, Gherkin is produced by the oocyte nucleus, that is, it is germline in origin. So, and torpedo is should be present in the somatic cell. So how was it proved? So how exactly it is proved that Gherkin is necessary in the germline and torpedo needs to be produced in the somatic cells? What exactly was done is they took 2 embryos. One was wild type, another in which there was another embryo was torpedo deficient. What they did is they then transplanted the pole cells, which will give rise to the germ cells in the future, and let the embryo develop. So the embryo in the top, it has a torpedo in the somatic cells, but not in the germline. And thus, when the embryo gives rise to its own, when the particular fly gives rise to its own progeny, then the progeny has torpedo defect in its germline, but the somatic cells itself has torpedo, where exactly so if we have seen, the follicle cells should have the torpedo, and follicle cells are somatic in origin. So where exactly the torpedo was needed, it is present right there. So there is no problem at all, and there is normal dorsal ventral axis formation. But in this particular fly, in which there was no torpedo in the somatic cells, but there was torpedo in the germline, we have the somatic cell, the germline containing can produce torpedo, but the somatic follicle cells do not produce torpedo, and thus there is no signaling between them and no dorsoventral axis formation. So the entire embryo is deventral essentially. So it obviously will be embryonic lethal. So these types of experiments were done in order to show that gherkin is necessary in the germline and torpedo is necessary in the follicle cells or the somatic cells. And as we have seen that we went through it, that nucleus first travels to the so anterior dorsal part of the entire embryo, and this only happens because of polarized microtubules. So essentially microtubules are equivalent to train lines that are present inside the cell, and various organelles can hitch a ride and travel to various parts of the cell. So nucleus does exactly that and travels to the dorsal anterior part, where it then deposits the gherkin mRNA and the entire dorsal ventral polarity thing happens. So the polarized microtubules are also important for the entire process to occur, and thus the options are, which are correct, are gherkin is present in the oocyte and polarized microtubules are in the oocyte are both are necessary for dorsoventral polarity formation in Drosophila embryos. Anyone wants to ask anything about this? Probably not. Okay. So we continue on the same topic. Which of the following is true in the context of axis specification in Drosophila embryos? Gherkin's protein is synthesized by the nurse cells and torpedo is synthesized by the follicle cells. Torpedo is synthesized by the nurse cells, gherkin is synthesized by the follicle cells. Bicoid mRNA is localized in the anterior, nanos mRNA is localized in the posterior, nanos in the anterior, bicoid in the posterior. Single option correct because it is a round type question. So this one is easy. Which of the following is true? So anyone wants to try and answer this? Okay, so this is something that I want to tell you all, that these sessions are designed to be interactive in nature. The only point is that normal NPTEL courses do not have this interactive component in which someone is talking to you and can help you clear out your concepts. And this is a great opportunity for you to actually talk about various problems you have during the course, various like questions you have, the queries you can address is during this particular session itself. So like interacting and at least like trying to answer the questions is great, I think, way to do that. So I would like encourage anyone to try and answer the questions. At least if they're wrong, even we can have a talk on why exactly is it wrong, and it will finally help you only. So we have two answers, which are C. Or bicoid mRNA is localized in anterior and nanos mRNA is localized in posterior. So with the entire discussion so far of axis specification, you know this is the correct option because everyone else is false. Gherkin and torpedo we have already seen. Gherkin is required in the oocyte, torpedo in the follicle cells. So both of them are wrong because they say nerve cells. And we have already seen the graph in which it shows bicoid is localized in the anterior and nanos is in the posterior. So This is another question on the dorsal patterning of Drosophila embryos, and we are asking which of the following is true. The enhancer for snail has higher affinity for enhancer of rhomboid for dorsal. So now we are looking at what exactly dorsal does after it gets into the nucleus in the ventral cells. The options are enhancer for snail has higher affinity than enhancer of rhomboid for dorsal. Rhomboid expression is restricted to mesoderm because the snail gene inhibits rhomboid. Mesoectoderm produces FGF8 receptors and mesoderm produces FGF8 ligands. Amnioserosa, which is the embryonic structure, is specified in the dorsal side of the embryo due to the presence of dorsal protein at the dorsal side. So let us directly look at the solution slide to it. So once dorsal gets into the nucleus, what exactly does it do? So this is the diagrammatic representation of the embryo. So if we go and have a cross section of the entire embryo, this will look like this, and we have the ventral cells right here, where the up to here I would say, where the dorsal has gone into the nucleus. So what happens is that dorsal goes and activates these three genes: snail, twist, and rhomboid. So we have the expression of snail and twist in the region where there is maximum dorsal. So a lot of dorsal is required for snail to be activated. Snail then goes ahead and inhibits rhomboid. So in the region where there is dorsal present but there is not snail present, only there rhomboid is activated. Here on, another thing happens is that where snail and twist are active, their FGF8 receptor is produced, and where rhomboid is active, FGF8 ligand is produced. Then again, what dorsal does is it inactivates these three genes: zirconate, toloid, and DPP, and thus these where there is no dorsal. So on the dorsal side of the embryo, only these genes are activated, and they form the various dorsal structures. So this is shown in this flowchart. Dorsal activates rhomboid, snail, and twist. Snail inactivates rhomboid. Snail then inactivates FGF8 ligand and does not form neuroectoderm genes. It inactivates. These are dorsal genes, so it will only form on this part. And then twist gives rise to FGF8 receptor, and then what? So FGF8 receptor is only present in this boundary, and FGF ligand is present in only in this boundary, and FGF receptor is present in here where there is snail expression. So from the diagram itself, rhomboid expression is restricted to the mesoderm because snail gene inhibits rhomboid, and amnioserosa is specified in the dorsal side of the embryo due to the absence of dorsal proteins at the dorsal side. So no dorsal protein is present in the dorsal side, and only there dorsal structures such as amnioserosa can be produced. So if anyone does not have anything on this, we will move on. The next question is, reciprocal transplantations develop according to how they would develop in their final position in the donor. This is because the transplant does not respond to the recipient's morphogen gradient and has a memory of the donor's morphogen gradient. Morphogen molecules in donor and recipient are different. Recipient organism does not accept transplant as its own cells. Transplanted cell's fate is pre-specified or intrinsic to it. The morphogen gradient merely induces its differentiation accordingly. So reciprocal transplants is something that we have already seen in the newt and if you remember the newt and tadpole embryo, when we transplanted the oral ectoderm part, so one formed the oral apparatus of newt. So the transplanted tadpole embryo had the oral apparatus of the newt, and the transplanted tadpole embryo, newt embryo, had the oral apparatus of the tadpole. No, so that transplanting the oral ectoderm part essentially transplanted the oral apparatus entirely between the two species. So the question is, how exactly this occurs? Is this because the transplant does not respond to the recipient's morphogen gradient and has a memory of the donor's morphogen gradient? Morphogen molecules are different. Recipient organism does not accept the transplant as its own cells. or the transplanted cell's fate is predefined, and morphogen gradient merely induces its differentiation accordingly. So again, this is a round type of answer, so only one of them are correct. Anyone tries to want to try and answer this? Okay, so let us look at what exactly is happening. So we have already discussed the oral apparatus transplantation, and we will now look into why exactly does it occur like this. So here we have the Fringe flag model, in which there is a fringe flag, and we see exactly how the gradient affects the formation of the fringe flag. Say there was a high concentration of morphogen on the left side of the flag, and the part of the flag which has the highest concentration can only form the blue color. So once till the concentration gradient is this high, it forms the blue color. But once the threshold, the concentration drop below this threshold, the concentration of the morphogen, then it will form white color. And finally, when it goes even low, it will form the red color. And that is why we have this three color formation. When the concentration highest, we have blue, middle concentration white, and low concentration red. So similar, this is what the entire French flag model tells us. Now, let us compare to another flag which is a bit complicated, the USS flag. Here, the same thing is occurring. So what they say is that, say there is a morphogen gradient which is occurring from the left side. Maximum morphogen gradient gives us the shapes such as this, the stars and stars on the blue background, and a low concentration gives rise, low to middle concentration gives rise to this alternate patterns of red and white. What we do is that we replace these two parts. This particular part of the flag from the French flag which had the maximum concentration to this part which has middle concentration. What happens is that this American flag part which gets deposited in the maximum morphogen concentration will form will develop into the part which had so say this particular portion is deposited here. It now experiences a very high concentration of morphogen. When it is shown a very high concentration of morphogen, it develops accordingly as it would have done in its own flag. So this particular flag portion, if it was on the leftmost where there was highest concentration, it would have formed the stars on the blue background. And thus, when it is shifted here and sees the highest concentration of the morphogen, it forms the stars on the blue background. Whereas this particular portion of the French flag, when moved to this middle to high portion, middle to low portion of morphogen concentration in the American flag, forms the vertical portion. So it acts as if it was this particular portion was here in the French flag instead. If you can understand where my pointer is. So if this was only transplanted between itself to here, it would have formed a white and the red portion because of the morphogen gradient. So even if it is transplanted to a completely different flag or there is interspecies transplantation, it will develop into it will develop according to the morphogen gradient according to the cells, according to the portion it would have developed into its own flag. So if this portion was here instead, it would have formed the red and vertical blue and red. But instead, even if it is in another flag entirely, it will still form the vertical blue and red, white and red, even though it is in the new flag entirely. So this is what exactly the morphogen gradient French flag model tells us. So here it is. An important feature of the model is that a piece of tissue transplanted from one region of an embryo to another retains its identity of origin. So since here in the French flag it would have formed a vertical white and red, even if it is transplanted, it still retains its original identity, but differentiates according to its new positional instructions. This phenomenon is indicated schematically by reciprocal grafts between the flags of US and France. So, the answer to this particular question is transplanted cell's fate is pre-specified or is intrinsic to it. So, retains its identity of origin. The morphogen gradient merely induces its differentiation accordingly. So, if this particular portion was shifted to the rightmost, it would have formed the vertical red portion only, and not a horizontal white and red stripes such as present in the US flag, is what the entire French flag model tells us. So anyone wants to ask anything from this? This is a bit complicated. I will ask you to actually go and read it up in Scott Gilbert, and if there is any problem, ask me in the next class. Anyone wants to ask anything from this? Okay. Which of the following statements are false? Is the next question. The phenotypes caused by mutations in maternal effect genes are observed in embryos that is homozygous for the mutant allele, but formed by a heterozygous mother. Free dorsal protein enters the nucleus in the dorsal region. Bicoid and caudal are observed in higher quantities in anterior and posterior side of the embryos respectively, and dorsal follicles do not produce spike protein due to presence of gherkin. So which of the following are false? Anyone will try and answer this? So I would probably discuss maternal effect genes in the next slide. So these three parts are already what we have already seen. So we can so this is more than one option correct. We can tick using our previous knowledge right here. So free dorsal protein enters the nucleus in the dorsal region, if it is correct or false. Bicoid and caudal are observed in higher quantities in the anterior and posterior side of the embryo respectively, and dorsal follicle cells do not produce spike protein due to presence of gherkin. So which of the following are false? Anyone will try and answer this? Okay, so we will for these three, we have already seen for free dorsal proteins enters the nucleus in the dorsal region. That is not correct, because we have seen that dorsal enters the nucleus only in the ventral cells of the embryo. Bicoid and caudal are observed in higher quantities in anterior and posterior side of the embryo respectively. This is correct because we have seen the graph itself, in which we have seen that bicoid is present maximum in the anterior and caudal is present maximum in the posterior. And finally, dorsal follicles do not produce spike protein due to presence of gherkin. This is also correct because we have seen that gherkin goes and binds to its receptor torpedo in the dorsal follicle cells and does not prevent the formation of pipe there, and thus only the ventral cells produce pipe, and the entire signaling is only happening in the ventral side, and that part only gets ventralized. So out of these, number 2 is the only one which is false. And now we'll discuss what maternal effect genes are. So we go into maternal effect genes because this is the only topic that we have not discussed yet. So during development, before the zygote is transcriptionally active, already maternally derived factors are deposited. So we have to understand that, as we have seen already, before the nucleus can in the dorsal ventral polarity part, we have already seen nucleus is only producing gherkin. But even before that particular production of gherkin by the nucleus, there are various mRNAs and proteins that are deposited in various parts of the embryo, which will give rise to this dorsoventral and anterior posterior polarity without even before the nucleus starts producing its own mRNAs. So as we have seen, bicoid and caudal, these are maternal effect, because bicoid and caudal are already present in the embryo even before the nucleus has started producing its own mRNA or proteins. and thus this is already predetermined from the mother's end. These factors, such as becoid and caudal, help in many important aspects of development, such as dorsoventral and anterior posterior polarity. These also help in production of the these also help in production of various other proteins such as Oscar, which help finally in giving rise to the final polarized embryo. So the asymmetry is produced by these factors themselves. Homozygous mutations in these maternal effect genes are not observed in the carrier female, but in the embryo produced by the carrier female in the next progeny. So this particular point is where we are discussing the Heidenberg screen that we will just see. So in the Heidenberg screen, I will just give you a short primer on it. So in the Heidenberg screen, we are trying to look at mutations that give rise to deformities in the embryos in the third generation. So we give rise to look at embryos which give rise to deformities in the third generation. This is what the Heidenberg screen is about. But in case of maternal effect genes, since these affect proteins or mRNAs that are deposited in the embryo, the third generation female will develop normally. But when it produces its own embryo, that is the fourth progeny, will have defects in it. So this is what maternal effect means. So this is the outline of the crossing schemes between the Heidelberg screen. So we do not look at all of this at the moment. We do not have to. This will probably be seen later, or we will probably go through it in detail later on. But as you can see in the F3, there is presence of the homozygous mutants. In the homozygous mutants themselves is where we want to see the particular defect. But if these defects have occurred only in the maternal effect genes, then in the F4 embryos need to be observed for phenotypic changes to find the maternal effect. So in the next generation, we'll be able to see from the homozygous F3 generation. And thus, which of the following statements are false? We have already gone through these three, and the phenotypes caused by mutations in the maternal effect genes are observed in the embryo that is homozygous for the mutant allele, but formed by a heterozygous mother. This is false because this needs to be a homozygous mother, as we have just seen here. So this is all we had for today. This reference is the Introduction to Developmental Biology by Scott Gilbert, the 9th edition. And this is the YouTube channel link on which the recorded videos will be uploaded. So if anyone does not have any questions, we can end the session right here. Anyone has anything to add? I guess not. So thank you everyone for joining for this week. I look forward to meeting you all next week. Thank you.